immune cell response and also the cell-mediated immune response. So it's basically a very long topic, but I'll try to make it simple but also understandable. Okay. So anyway, to review, innate immune system is the one responsible for eliminating most of the pathogens because it's our first line of defense. So there were 5 basic mechanisms we discussed last week. So those are your microbial sensors, your cellular components and phagocytosis, your natural killer cells, complement system, and your mediators of inflammation and interferons. So now let's go to adaptive immunity. So your adaptive immunity is actually your second line defense. It is actually highly specific. It means that it is very specific to a particular antigen, and that it also responds to that specific antigen. Okay, so it's specific because it has a component or a property called immunologic memory, and because of this immunologic memory, there is actually a rapid response of that immunologic response to the offending microorganism. Okay, so there are basically two types or two mechanisms or processes under your adaptive immune system, and this is your cell-mediated immune response and your antibody-mediated immune response. So, do you have any idea what is adaptive immunity in your own way or in your own words of understanding? You can raise your hand, please. I'll call somebody. As much as possible, we will have a very interactive discussion so everyone can participate. Or who will volunteer? Okay, so if no one will volunteer, I'll call randomly. So online, I will call online, okay? Don't worry, you'll be next. Okay, I can ask attendance here, please obey. Okay. So, who is Apichayai? Who's this? Apichayai, let's see the whole name, sorry. Oh, this one. Who is Abaya Das Vishnudat? Online class. Who is Abaya Das Vishnudat? Can you answer my question? What is adaptive immunity for you? Hello? Okay, I'll call another. You better be present, guys. Abiraj Sin Vijay Sin. Who is this? Still in online. Still online.
Hello.
Cannot see your face. Abiraj, are you present? Like virtually present? Hello? Abiraj Sin Vijay Sin.
Yes,
Sige, last call from the online. How about
immunity.
Anjali Anil Dibase?
Adaptive immunity.
Anjali Anil Dibase should be around because you're famous here.
Hello, Doc. Hello, Doc. Can you hear me? Hello, Doc.
Why are they not answering, man?
Hello, Doc. Hello.
Okay, so I think if the online class is not answering, I'll better call the face-to-face student here. Huh? Okay, volunteers. Okay, I will give points to those who answer questions. Always not all your okay, and especially the online because it's really very hard to have a mixed type of teaching because we also need to interact with those online. Okay, so we want to make sure that they understood, they really physically or virtually present, not just listening. Okay, okay, so let's be mindful of our responsibility as a student. Okay. So anyway, who will volunteer? Okay, you're the first one who raised hand. What's your name? Akshaya. What's your name? Akshaya. I cannot see. Okay, you are alumni. What's your laboratory group later? Two. Your group two. Okay, thank you. Sige. So what's adaptive immunity? Immunity that we get adapted once we're exposed to an infection or even example measles. Once we're exposed to it, and then we develop get adapted infection, and after that, only from then we get adapted to that particular kind of antigen. Okay, very good explanation. So the keyword there is antibody, and also the keyword there is adapt. Okay, so it means that there's already beginning memory of the previous infection. So once you are once on your second encounter, it won't be the same as the first encounter. Like probably there will be no recurrence of the infection, like the measles or chickenpox. But the other one is second is if it goes like it worsens, no, or probably it gets milder. Okay. They're telling that they're answering, but we did not hear. Maybe the mic. Oh, okay. So the microphone. So okay. I think my bad. So how am I going to interact with them? Maybe because you are speaker. Speaker? I know the laptop. Yeah, the one. This one.
Or else you can just ask them to write.
No, it's here. I think you can hear them now. They can control volume. Can I have them
Or else
to
you
speak?
can just write their answers in the
Yeah.
chat section.
Can you speak there online, class? Hello? Or they can do in chat
No,
box.
no, but it won't be.
Yeah, they're answering.
But you won't hear that.
Okay, so
So
try.
you can just ask them to write their answers in the chat box.
Okay, someone just be on your guard. If there will be answers in the chat box. Okay. So anyway, let's go back. So in other words, adaptive immunity is more on antibodies. Okay, memory cells, and they are also actually the cell types there are your B cells and your T cells. So the first thing that we're going to discuss about adaptive immunity is their cellular basis. So where are they coming from? Okay. So first things foremost, they are lymphoid cells. No. Do you recall our hematopoietic stem cell discussion last time? It is divided into the hematopoietic stem cell will differentiate to myeloid and lymphoid cells. Okay, so let's focus on the lymphoid cells because there's where there's where the adaptive cell or adaptive immune response will actually act on. Okay, so lymphoid cells play a role in the adaptive immune response. So the hematopoietic stem cells will reside in the bone marrow. So you can see the picture. So this is actually your mother cell. Okay, so these stem cells will destined to become your B lymphocytes. So they develop in the bone marrow and T lymphocytes. Or once they migrate to the thymus, they will mature. Okay, so let's first talk about B cells. So these B cells migrate to secondary lymphoid organs. So what are your examples of secondary lymphoid organs? Okay, very good. Spleen. Okay, and another example. Okay, another. So I think the major is spleen and your thymus, okay? But mostly, predominantly, your B cells goes to your spleen and then they are activated by an encounter with the antigen and then becomes your plasma cell. So these B cells, once they encounter antigen, in our blood, they become or they mature to become your plasma cell. So they will be the one who will secrete antibodies later on. Okay, so just remember the concept. So how about your T cells? Your T cells are your lymphocytes. Okay, they are produced also in the bone marrow, but they travel to where? Okay, so they go to your thymus to mature. Okay, so once they go to the thymus, what happens there? What happens there? They go there. What? How do they mature? Okay, they mature through differentiation. Okay, so actually they mature based on the process of selection. Okay, but we'll discuss it later because these T cells will then become matured soldiers, no, with specific types of receptors that they will be functioning. They will be functioning. Okay, so these T cells form subclasses. So these major subclasses will be your CD4 and your CD8 T cells. So these are the major subclasses of your T cells. Okay, so they are actually your T cell is actually called your cell-mediated immunity. They are the cells or lymphocytes responsible for your cell-mediated immune response, while your B cell will be the one responsible for your antibody-mediated immune response. Okay, so these T cells and your B cells will be discussed no further later. So for this time, we're going to discuss first antibody-mediated immune response. So these antibody-mediated immune response, one example is your CD4 T cell. Actually, your antibody-mediated immune response, it's usually your CD4 T cells that usually acts on the antibody-mediated because they are the ones who will portray in giving signals to your B cells to express antibodies. Okay, while your cell-mediated, usually they are the ones your CD4 and your CD8. So let's discuss first in your antibody-mediated immune response. So your antibody-mediated immune response, it is your CD4 T cells that will recognize your MHC2 or your class 2 MHC molecule. So what is this class 2 MHC molecules? I'm discussing this, preempting you about the MHC. They are the ones responsible to present them to the T cells. Okay, so they are molecules that actually present in your T cell receptors to be the one to be presented. No, it's like what do you call it? Antigen presenting cells, like that. So they are actually on the surface of it. Okay, and the consequences of these interactions, actually, your cytokines will be the one producing or stimulating your B cells to express the antibodies. So this is actually the explanation of your antibody-mediated immune response. So again, your CD4 T cells will interact with your antigen via binding to class 2 MHC molecules, and by this time they stimulate, they stimulate the cytokines to let the B cells express antibodies. So this is your antibody-mediated immune response. Okay, so your B cells, once they got an encounter to the antigen, they then mature to become plasma cells. Okay, so that's it. So your cell-mediated immune response. So your cell-mediated immune response. What is the difference between your antibody-mediated immune response and your cell-mediated immune response? So this is the difference. So your antibody is more on the CD4, right? More CD4 stimulating your B cells to produce antibodies, while your cell-mediated immune response actually has both your CD4 T cell functions and your CD8. So the CD4 again still stimulates B cells to produce antibodies, and that's what you call your delayed hypersensitivity, no? And how about your CD8 T cells? It actually causes direct destruction, no? By binding to your MHC class 1. So those are the difference between your antibody-mediated and your cell-mediated immune response. So for your cell-mediated, it has two types of T cells involved, okay? Your CD4 and your CD8. Your CD4 binding to your MHC class 2, no? And then stimulating the B cells, while your CD8 is binding to your MHC class 1, and then has the potential to directly destroy the pathogen. Okay, so do you understand the concept? Just tell me if you haven't, because this is very important, especially when you're going to internal medicine later on, or you'll be exposing yourselves to become immunologists, no? So we have to know all these things. So again, this is actually a picture. No, I got this from Google. And humoral is actually your antibody-mediated immune response, while your cellular immunity is your cell-mediated immunity response. So your humoral is actually the popular cell involved here is your B cell and the antibodies, and then the CD4 T cell here. And then your cellular immunity, it's both your CD8 and your CD4. And then actually this becomes specifically your cytotoxic T cells, okay? So another concept that we want to discuss is about antigens. So what is antigen? Okay, I'm gonna call what? I want raise a hand or an answer to the from online people. So I will be calling Afrin Darbar. Afrin Darbar. Hello. Just write in the chat box if you're not heard. Hello, Afrin Darbar. Are you virtually present?
Yes, doc.
Okay, very good. So what is antigens, or what are antigens? What is an what?
A substance that react with the antibody, doc.
Okay. In example of antigen. I mean, example of what do you call it? Example of the reaction, or that substance that reacts to the antibody. Example. Okay, so Afrin is actually right that it's a substance that reacts with antibody. But one example of this are your foreign cells. Example, your the virus, bacteria, or even immunogens, any immunogens that causes reaction, no, to your body. Okay, so what actually causes immunogenicity? Why is it that some particles or some substance reacts immediately to antibody? It's because of these five. Okay, thank you for that, Afrin. So anyway, five properties for immunogenicity. What are those? Recognition of foreignness, size, chemical and structure. Actually, these properties will depend on, these properties actually, it's where the immunogenicity is depends upon these five properties. Okay, so for example, recognition of foreignness. So if our immune cells recognizes that you are a foreign cell, so that actually causes reaction. No? So that's one property. Number two is for the size. No, so the larger and the more complex size of your immunogen or antigen causes a reaction to your or causes immune response. Okay, so that's another property. The third property is your chemical and structural complexity. So it actually depends also if it's a homopolymer or it's just a one single substance or a complex or a heteropolymer. Okay, it has so many various genes involved, so it causes it's actually immunogenic or it causes a reaction, to promote immune response. Okay, so another property, genetic constitution of the host. So it depends on the genes, no, of your genes. For example, if your genes actually is or your genetic predisposition or family is very susceptible to to any infection, so you're also very susceptible. So like that. So that's another property that their property of immunogenicity. And the last property is your dosage, route, and timing. So it depends on the concentration of the antigen administered to your body. So if it's so many, like overdose of so many antigens encountered or introduced to you, so of course you will be having a reaction, and it probably is very severe, no? So that's the property. These five concepts are actually your properties of immunogenicity. Okay, so these another concept to remember is that these antigens are also the ones we call antigen recognition molecules. So they actually there's a system, no, that our body um, is have has the potential to have a recognition system. So that's because of the immune system that they immediately can detect if whether it's a self or a non-self cell. Okay, so that's it. So major histocompatibility complex. So this major histocompatibility complex, or MHC, as what we all know of as molecule that actually the one presents the cell, no? I mean the one that is attached to a cell that can cause that can make it a marker to present it to the T cells, no, to detect that this is a foreign cell. Okay, so those are called your MHC or your HLA complex or cells. So actually, there are three classes of MHC: your class 1, class 2, and class 3. But the most studied are actually your classes 1 and 2. So these molecules are bind peptide antigens, and they present them to the T cells. Okay, so they are actually these MHC are actually in your antigens present there, and then they are presented to the T cells. Okay, so there's also a receptor of your T cells that so they are actually responsible for T cell antigen recognition, and they play a significant role in controlling immunologic functions. So, as what I've discussed, this is actually from Jawet's. Class 1 and class 2 are most studied. So, these table summarizes the difference between your class 1 and class 2. So, just remember that their cell distribution is actually different. Most of it is from class 1. Most of them are nucleated, coming from nucleated somatic cells. Those are class 1 MHC. While your class 2 usually from your presenting cells or your first line defense, your macrophages, your dendritic cells, B cells, and so on and so forth. And for your class 1, where do they bind? They bind to your CD8 T cells, while your MHC class 2, they bind to your CD4. So just remember, CD4 class 2 and your CD8 is class 1, okay? So they should match, no? And they should actually be partners, no? Because they will be the one, if that particular T cell is a CD8, then they should bind to the class 1 MHC molecule and not on the class 2. No, they don't bind to class 2, just on the specific class 1. So the sizes actually, the larger molecule is the class 2, which has 10 to 30 residues. Okay, so another is that we discussed the MHC class 1 is that most of them are nucleated somatic cells in the body, no? But except the retina and brain. Okay, so these major histocompatibility complex actually controls your immune responsiveness. No, so the more it is highly expressed, no, the more your immune response is very sensitive to the pathogenic organism, okay, or to the offending organism. So your MHC classes 1 and 2 genes exhibit your genetic variability with high degree of polymorphism. So what does that mean? Your MHC classes 1 and 2 actually has variable genes, no? So it depends also, that means that it's a polymorphic, no? So MHC polymorphism actually has different peptides, no? And that causes its polymorphicity, okay? So it means that it's variable from one person to another, so it's different, okay? I mean, it possesses a genetic variability. So MHC complex, what's the structure of it? It actually composes of your alpha helices and your beta pleated sheets. So do you recall your chemistry class that the structure of, I think this is level 3, tell me if I'm not mistaken, that this is actually a level 3 complex of structure, chemical structures, atoms, diba, sa figures sa chemistry. And they consist of your alpha helices and your beta pleated sheets. So the floor shape of your MHC composes of the beta pleated sheets. So they are very highly complex. While the sides of it, the clefts, are actually your alpha helices. So another concept to discuss is the antigen processing and presentation. So these antigen processing and presentation, these antigen processing and presentation is the hallmark of the adaptive immune response. Okay, so these antigens with self-MHC molecules present to the T cells with appropriate receptors. So that's what we mentioned earlier, that if it's a CD8 T cell, it is usually the MHC molecule responsible for it is your, if CD8, what's it? What's the, what? Two, okay? While how about your CD4? What? Okay, so your CD8 is your, okay, and while your CD4, okay, so just remember that. So for the protease, so cytosol, it is actually the antigen has these antigen presenting cells has the components, your proteasome, which is a cytosolic proteins that is broken down by the proteolytic complex. So they actually process proteolysis. So some antigens are called super antigens. So what does that mean? Your super antigens actually are proteins that examples, your bacteria and viral antigens that are able to activate large numbers of T cells, causing a cytokine storm. So these actually causes enhancement of your cytokines to what they call this, to be activated, no? Like, I mean, there's overproduction of cytokines that causes a storm. So these actually these bacteria or viruses are very toxic. They're very kind of causes death. No, they have higher mortality rates. No, because or morbidity and mortality rates because of this property that they causes overproduction of cytokines. So example of these super antigens are your staphylococcal enterotoxins, your toxic shock syndrome toxin, and your group A streptococcal pyogenic exotoxin. So your next topic is your B cell and your B cell and antibodies. So B cells actually have homogeneous clonal immunoglobulin. So these B cells possesses homogeneous, meaning there's only one immunoglobulin present on the surface. Okay, so immunoglobulin serves as the B cell receptors. And these B cell receptors are called your humoral immunity. Actually, it's where your antibodies are present, no? Or it has the immunoglobulin. Okay, so immunoglobulin serves as receptors or B cell receptors. So these immature B cells actually carry IgM on the surface. So all of the B cells that are still not matured to become plasma cells has IgM on its surface. So B cells form clonal selection as antigen binds to the BCR. So these B cells actually have the clonal selection. So it means that there's expansion of these antigen that binds to the B cell receptor, causing differentiation to become plasma cells. Hence, they secrete antibodies. Okay. So, for example, in this illustration, the naive or the immature B cell has recognized an antigen. So it then stimulated it by releasing some of its cytokines. And then this naive B cell then becomes your activated B cell. So once they are activated, they possess clonal expansion or selection as it binds the antigen to the B cell receptor. So once this has begun, there's actually it has a higher affinity to all of these immunologic properties, hence differentiates to memory B cells. Okay, so these memory B cells are also called your plasma cells. So they already have that antibody present in them. Okay, so where do they differentiate? So they differentiate in your germinal B centers to become memory cells. So another concept, antibody structure and function. So antibodies are actually immunoglobulins, okay? So they compose of your 20% of your plasma proteins, and these antibodies are formed by different clones of cells. So this clone expresses antibody capable of reacting with different antigens. So this clonal expansion actually can be polyclonal or monoclonal. So it depends on its description. Like if it's polyclonal, there is actually reaction to different antigens, while your monoclonal only reacts to a single clone of cell. So that's how expansive is your immunoglobulin. Okay. So immunoglobulin, there's a structure. You should be able to familiarize the structure of your immunoglobulin. So these are the parts of your immunoglobulin. So it has two identical light chains and two heavy chains. So can you see the slide? I mean the picture. So the picture actually this is the an example or a structure of your anatomical structure of your immunoglobulin. So it composed of your light chain, which is this one, two light chains and two heavy chains. Okay, and then these light chains can be has two subtypes, but only one can be linked with this. No, so it's a kappa light chain that it could not be both. So it's either kappa or lambda. So the two subtypes of light chains are your kappa and your lambda, while your heavy chains, this actually has the distinction of classifying them to five immunoglobulins. So the first one is your gamma, okay, alpha. Actually, these are alpha, delta, epsilon, gamma, and mu. Okay, these are your five Ig classes. So you see the amino terminal portion of the light chain. This one here. This is the amino terminal. Okay, can you recall your chemistry now? So just try to remember them. The amino terminal is the one that's distal to the distal to your to this structure. Okay, so this is here in the end. While your carboxyl here more proximal, no, to the base. So anyway, amino terminal actually is the portion of the light chain that contains the antigen binding site. So this is where the antigen binding site is located. Okay, so both light chain types can actually occur in all immunoglobulins, but only contains one immunoglobulin of each light chain. So that's what we try to emphasize that kappa, only kappa, no light chains has the one that has both, no? Only if it's kappa, then it's kappa. If it's lambda, then it's only lambda. Okay. For your heavy chains, so your heavy chains, the amino terminal of your heavy chains, this one here, no? This one here, the activity here of their amino terminal is actually your antigen binding site also. So this is the Fab portion or the Fab fragment of your immunoglobulin. So actually, the whole part of this, the Fab fragments are your antigen binding sites. Okay, while you're here in your FC fragments, which is your constant region, actually it's where the biologic function of your immunoglobulin takes place. So example, cytokine activation or destruction, like that. So there's where the biologic activation takes place in your FC fragment. For your antigen binding site, it's in the Fab fragments. No? So both your light and heavy chains of an immunoglobulin are subdivided into variable and constant. So this is here in the picture. Both of them has variable, constant, constant. But the difference is that your heavy chains has more of the constant regions than your light chain. Okay, so that's the structure of your immunoglobulin. So another picture. This is a more simplified picture of your light chain plus your heavy chain. This is actually the heavy chain here, light chain, then the variable region and the constant region. Okay, so both are actually linked by the disulfide bonds or bridges. So for your antigen recognition molecules, so they are both subclassified into five, right? So what are those? Your IgG, IgA, IgE, IgD, and your IgM. So for your first is your IgG. What is IgG? Those are major classes of your immunoglobulin. They're actually present and predominant in the serum. Okay, they're the most abundant immunoglobulin in newborns. Why? Because very good. Okay, who answered that? Because because it is the only immunoglobulin that process the placenta. So just remember IgG, placenta, and you can cross placenta. Okay, so that's why it's most abundant in newborns. And it has a subtype. Okay, so your IgG1, IgG2, IgG3, IgG4. So your IgG1 is the most common or most abundant. They compose of 65% of the total IgG in our body. And your IgG2, it's against any encapsulated bacteria. Your IgG3, it is effective in complement, yes, activation. Your IgG4, it does not involve in your activation of your complement system. So your IgG3 is the one involved in your complement. Your IgG4, it is more against on your encapsulated bacteria. IgG5 is the most common or most abundant in of the total IgG. So another one is your IgM. So your IgM is the first immunoglobulin produced in response to an antigen. So that's example of this is when the patient has a dengue fever. Now, why do we need to get both the dengue test IgG and IgM? Why do you think so?
Recently, when the IBM
Sorry? Because of this explanation. Because it is the first immunoglobulin to be produced in response to an antigen. So you need to get both so that you will know that his dengue fever is acute. So it means today, no, it is actually you are having an active dengue fever, no. No, if it's already negative, if it's non-reactive, if your IgM is already non-reactive, but there is your IgG reactive. So it means there's it's recent. So probably recently or in the past you have that dengue fever before. Okay, so that's the difference. Okay, so that's why we need to get both the IgG and the IgM. Even in COVID infection, we also get both the IgG and the IgM because we want to know if it's acute or it's already before, it's like a recent infection before, and then that's why he has that IgG, okay? So more or less, that IgM actually is the first one to be produced to respond to an antigen. So it is also the most efficient, no, immunoglobulin agglutination, complement fixation, and antigen antibody reactions. And it has the highest binding capacity and cross-linking of all immunoglobulins. So of course, because you're the first one to be produced, so you should be the one to have the highest binding capacity, right? You have to attract a lot, you have to make sure that you induce a lot of mediators or inflammation or anything that can attract neutrophils, macrophages to that side. So that's the purpose of your IgM. And actually, the presence of your IgM in the fetus, it only signify that you have an intrauterine infection. So example, because IgM should not be in the fetus, right? So once it's present in the fetal blood or newborn serum, then probably that fetus or that newborn could have had her mom or her mother might have an intrauterine infection. That's why. So another IG class is your IgA. So your IgA, it is actually a major immunoglobulin for mucosal immunity. So more or less, um, more on the glands, more on the lining glands, more on the mucous membranes. So that's your IgA. So examples, where does your IgA go or is located? Where? Milk, saliva, tears, respiratory, GIT, GUT, and any secretions, no? Any extravascular secretions, because IgA should not be present intravascularly. No? It should be more or less on the secretions, that or on the glands, linings, and mucous membranes. So it is usually the immunoglobulin present because it's part of your first line of defense against bacteria and viruses. Okay, so if you're given a question or an example in the exam, patient with having what's the what's where what immunoglobulin is responsible for or what immunoglobulin is present in the saliva. Okay, if you're asking those, you've been asked by you've been asked by your professor to answer, so that's your IgA. Another immunoglobulin, IgE and IgD, actually are not that gonna cause any significant details, but the most important thing that we need to know about your IgE is that it is found in your mast cells, basophils and eosinophils, and it triggers allergic responses or of immediate type, like anaphylactic shock, like that. So it's your IgE that is responsible for that. Another is your immunoglobulin D. Also, only in trace amounts of this immunoglobulin, but it's more on the mature B lymphocytes that have not yet encountered antigen. So, okay, just remember all the important characteristics or features of each of these immunoglobulin. So in this table from Javas, it summarizes the differences of or the characteristics of each of these immunoglobulins. So these are the heavy chain symbol of your immunoglobulins. For the molecular weight, it's more on the heavier is your your IgE. Okay, then the second one is your IgD. And for your, for the serum half-life, your IgG has the longest half-life. No wonder why we get IgG and then we still determine its presence even for several weeks, diba? Even if the infection is already gone, because it has a very long half-life, like 21 days. So this one, just remember this. It's the only immunoglobulin that process your placenta, your IgG. Okay? So this is how you will gonna read tables. You need not to memorize all this. Just understand them. Like what should I make use of this table? No? Like for example, this one. You remember this that the IgG is only the immunoglobulin that can transfer to the fetus. Okay. Anyway, next slide. Immunoglobulin genes and their generation of diversity. So, your immune response actually there is very diverse. No, it generates a lot of antibody specificity. So that's your because of your somatic recombination. So a lot of these areas like there's multiple gene segments, combinatorial association, random combining of different L and H chains, somatic hypermutation, and junctional diversity all contributes to this diversity. So that's your somatic recombination. So for immunoglobulin switching, so actually this is what happens. Initially, your B cells is bound to your antigen, and they carry IgM specific for that antigen. Okay, but later on, there's this immunoglobulin switching that's happening. Okay, because of gene rearrangement. No, your antibodies will generate this gene rearrangement, okay, causing this antigenic specificity to be of different Ig class. So, for example, the IgM, it causes switching to become IgG. So that's actually because of these interleukins, your cytokines, your interferons, all of these are the ones responsible for the immunoglobulin switching. So for antibody responses, so there's primary and secondary responses. For the primary response, so this is the primary response. This is when an individual encounters an antigen for the first time, and then the antibody is produced in response to antigen. So it is detectable in the serum within days, no, or weeks, then declines for several weeks. So that's the primary response. However, for the second response, okay, so the second response, so once the individual encounters the same antigen on the second time, no, with this in the same month or even years after the primary response, it is more rapid and has higher levels and it is much longer. So it means that if you develop a secondary, your body is trying to cope with that same antigen, no, by performing the secondary response mechanism. So once you are encountering that antigen for the second time, so it is more rapid, it generates higher levels, and it persists much longer because of this memory or immunologic memory, okay? Because your cells tend to develop antibodies causing memory cells, no? They remember that antigen. That's why it's more rapid, it's faster, they can react faster to the infection, and then they actually can generate higher levels of that T cells, no? And also higher levels of that immune response, and it can persist much longer. Okay, that's also the mechanism why we need to be exposed, or for example, to be vaccinated or boosted, because we need to have another second encounter of that organism. Or, for example, if you are already having dengue, okay, example lang, you have dengue and then it's your second time, so your body actually will react to that antigen more rapidly, and it also generates higher levels. So a second dengue might be a very dangerous one, okay, because of this secondary response mechanism. So one responsible of this secondary response is your antigen-sensitive memory cells, because it binds more firmly to the antigen. Okay, so that's the mechanism. So in this diagram, show it is in here in this diagram that the primary response is here at this level. But once you have secondary infection or secondary encounter to the same antigen, it generates higher levels. So it's more rapid, it's more efficient, and it's longer, okay, than the primary response. So these are the protective functions of your antibodies. So no, I think we all know this. It enhances phagocytosis, it neutralizes viruses, also the toxins of the viruses or bacteria, they have complement-mediated lysis, and there's antibody-dependent cell cytotoxicity. So forms of immunity. So let's differentiate between your active and your passive immunity. So what is active immunity? Do you recall active immunity? Kumaragorosomia here. Kumaragorosomia. What's active immunity for you? Okay, I'll call number Patel Akshay Kumar. Where is Patel Akshay Kumar? Okay, so what's active immunity? Sorry. Okay, the infection. Okay, first time encounter of the infectious agent. Okay, and then you develop immune system. Okay, that's good. So example of active immunity. Okay, need those. Next, what else? Online. Online. We're almost done. Anyway, I think nobody is answering. So active immunity, it actually is the one that when an individual comes in first time contact with a foreign antigen, it develops immunity, with that. And example of this are if there's a immunization of live vaccines, exposure to microbial products, and transplantation. So example, your measles, chickenpox, no? And it is actually the individual that produces the antibodies, no? It's immunity, it's natural immunity, and it's actually long lasting, okay? But the adverse effect or adverse outcome of this is some infections actually can also trigger formation of circulating immune complexes. So it actually generates complications, no? Or develops complications like glomerulonephritis, rheumatic heart disease, okay, rheumatic fever. So also it can cause what? Yeah, more on autoimmune responses, okay? Because of that severe infection. Usually, this is the adverse outcome of having an active immunity. While passive immunity, it is the administration of preformed antibodies. Okay, so that's where the vaccine is is introduced, okay, to the host. So recipient receives large concentration of antibody immediately, and then your body reacts to that antibody given. But it's not long term. That's only the disadvantage because it does not possess long term protection, and it is only indicated when patient has no time to produce antibody response. So, for example, very good rabies, no? So you don't want to wait patient to get immunized and then the patient is already having that serious infection. No? So you need to give rapid anti-rabies na immunoglobulin to have a immediate effect of the antibody response. Okay, but the adverse outcome of this is that it's common to of these individuals to develop hypersensitivity reaction. So that's why when we get vaccinated, we usually monitor them with the reactions to the vaccine. If you are hypersensitive with it, okay. So another example of this is also hepatitis, right? Hepatitis B. If you got a needle stick injury and the patient had a hepatitis B infection, so of course you need to be injected right away, so that to protect yourself. Okay, so that's passive immunity. So for the T cells, your T cells is actually is more likely involved in your cell-mediated immunity response. So how T cells develop is also from hematopoietic stem cells. It matures and undergoes differentiation in the thymus, and they become committed cells by expressing specific T cell receptors. So what were the T cell receptors being discussed? The CD4, K, and the CD8. Okay, so this is what I am talking about lately. So your T cells during the differentiation stage in the thymus, they undergo selection, okay? So in positive selection, the cells recognizes the self peptides and the self MHC with weak affinity. So during the positive selection, these type of cells with weaker affinity will survive. Okay. Well, during the negative selection, so those that were evicted, so the T cells that are evicted because of this abnormality, self-peptide and self-MHC with high affinity, they are actually killed because they are potential to develop um, autoimmunity. Okay, like they're going to kill or eat self MHC or the self cells. Okay, so they're going to eat the normal cells. So those are the ones that should be killed. So that's how the selectivity or the selection is going through the process for your T cells. So once these cells survive, they become matured into either CD4 or CD8. So that's where they will be classified into CD4, CD8. So after their selection, okay. So if they see that if you have a higher affinity to kill the self cells, then you should be addicted, no? So that's the function of your thymus, no? They undergo differentiation, maturity. So once they mature, they differentiate into either CD4 or CD8 cells. Once these cells determine that they are not going to kill the self cells, okay? So those are the this is the process of cell maturity, and then how they undergo differentiation into CD4, CD8 cells. So another is the structure of your T cell receptor. The classification or the types of your T cell receptors for each of these antigens. So The class cells are both can be alpha beta T cells or can be gamma delta T cells, but mostly the majority is from the class of alpha beta T cells. So these class actually is subdivided by either CD4 or CD8. So this should be remembered always. Your CD4 T cells, your MHC class 2, it is bound to your MHC class 2, while your CD8 is bound to your MHC class 1. Okay? So also same as with your B cells, your T cells also have constant region, variable region, but the only difference is that it has an invariant of CD3 complex region or portion. So because it is responsible for transducing signals to received by your T cell receptor when your antigen is recognized. Okay, so these are the parts or the structure of a T cell receptor for antigen. So this is the difference or the similarity of both the T and B cell receptors. So for your B cell, it has still your light chain and your heavy chain, also has the variable and the constant regions. And then your T cell also has a variable and constant region, okay? But there is no light chain and heavy chain, only the variable and constant region has remained. But the difference also is that your T cell also has the alpha chain and the beta chain, which is the most common, diba? The alpha beta here, alpha beta T cells is more or less this class has the majority of present in our body. Okay, this kind of T cell receptors. Okay, so just familiarize the structure of your B cell receptor, your T cell receptor. Why does the B cell receptor has light chain and heavy chain? Because your light chain, that's your antigen binding site, diba? Your heavy chain has the immunoglobulins. Okay, that's why the structure is more complex. While your T cell structure is just simple, only variable and constant region, plus the invariant of CD3 complex, which is responsible for these transducing signals. Okay. So for responsible, the mechanism of proliferation and differentiation of your T cells. So there are two signals present for to activate your CD4 T cell. No? So the first signal is the T cell receptor on the T cell will interact with the MHC, MHC assigned of it, okay? So presented on the ATC. So the second signal is actually through co-stimulation. Because this co-stimulation will the responsible for this co-stimulatory molecules are your B7 family co-stimulatory molecules. So they help in activating your CD4 T cell. No? So that's one mechanism for your CD4 T cell to be activated. For your CD8 T cell, it doesn't require any of the co-stimulation at all because it actually has the potential to directly destroy a pathogen. So it only has this signal. So once this CD8 T cell is activated, the cytotoxic T cells will only produce the interleukin 2, interferon gamma, and your growth differentiation factors, your transforming growth beta factors for your T cells, which is responsible um, uh, to kill the pathogen. No? This CD8 T cell only interacts with the MHC class 1. For the CD4, it interacts with your class 2, plus it involves in this kind of signal, your co-stimulatory signal. Okay, so for your T cell effector functions, so your CD4 effector cells has these subtypes, no? So your TH1, your TH2, TH17, and your T regulatory cells. So for your TH1 is actually triggered by your interleukin and your interleukins 2 and 12 and your activated macrophages. So it causes B cells to produce IgG, interleukin 2 and interferon gamma, and it involves bacterial clearance by direct destruction. So actually the function of your Th1 is more on direct destruction, no, of the microorganism. While your Th2, its function is more on the activating your mast cells and eosinophils. So what's the difference between your Th1? It activates your, or it triggers your activated macrophages. For your Th2, it activates your mast cells and eosinophils. Okay, so these activation causes the B cells to synthesize IgE. No, then the rest are history. So there's cascading cytokine or inflammatory mediators, hence the production of your interleukins, no, the 4, 5, 9, and 13. So other than that, it is this stage 2 is also responsible in response to the helminths, no, aiding in response to helminths. So any helminthic infections, it's the Th2 that is responsible for that, okay, in killing those parasites, okay. So the Th2. So the Th1 usually more on bacteria, diba? Bacteria, Th1, Th2 more on the helminths, parasites, so on and so forth. So for the Th17, this one, actually, DTH17 is more responsible in inducing a lot of inflammatory mediators. That's why there's this production of your interleukin 8, 17, 21, 22. But the most important interleukin or inflammatory mediator for this stage 17 cell is that this interleukin 8, no, the presence of the production of this interleukin 8 can actually become the potent chemokine. What's the function of your chemokine? It attracts, no, it attracts or it attracts a lot of neutrophils, no, to recruit for microbial invasion. I mean, fighting for against the microbial invasion. So for the Th regulatory cells, they are responsible for suppressing the T cell response. So actually, these Th regulatory cells, they suppress the immune response, no, in general. So they help in lowering down or mellowing down your immunity, your response to the pathogen by these Th regulatory cells. So actually, these Th regulatory cells are actually made of because of the exposure to these growth TGF, beta, and interleukin 10. Okay, so another T cell effector. So we already discussed about CD4. So the next effector cell we will discuss is your CD8 effector cell. So this CD8 effector cell is actually, since it is a direct, its focus is more on direct killing, no direct destruction. So it's called also your cytotoxic cells, okay? They directly kill infected cells, okay? They don't undergo all the process, no, sa CD4, because the CD8 actually is direct, so it's cytotoxic. There's no co-stimulatory na, diba? So that's your CD8. The method of killing of these, the method of killing for these CD8 effector cells, or the method of killing of these CD8 effector cells to the pathogens is through its granules, no? So what we call these granules or granzymes are your perforin and your granulysin. So they actually induce apoptosis, no? Once they enter the infected cell. Okay, so I think that's all. Do you have any question? For the complement system, it's a very separate topic. So I will be giving you an e-learning video. I will be lecturing on complement system through an e-learning video, and then we'll be presenting it to class next week. Okay. So for now, let's be fun. Let's prepare for the lab activity later. Okay, I think it's 3 o'clock. So do you have any questions? Did you understand? Any clarification? So I think at the end of the lecture on immunology, we will have a very long quiz for this, but we'll try to coordinate it with our department.
So are you uploading it in our file section? Are you uploading this in our file section? You will be uploading the PowerPoint and file.
No, I will not be uploading the PowerPoint, but I will be giving you my notes. No, I made a notes or summary notes for my lecture so that you can be able to review it. Okay. But if you have anything you want to clarify, just message us so we can explain. Okay. But the source of my lecture is actually in Java. It's not from Java, so you can actually the explanation is there.
Itong inyong notes. Sige, start na lang. Kami na live po sa ilang classroom lang.
Sige, sige. Regarding the e-handouts that I will be uploading, we will be distributing it, no? But they will be the one in charge, the junior MEUs. Thank you. And the word, if there's in the laboratory, I will remove that activity right away.
Didn't hear anything. To the online students, yung activity will also start at 3:15 since the demonstration will start by then. Please make sure to be in this wall where they have by 3:15 because your professors, your faculty members will be there with you, and then after the demonstration, you'll have your separate meeting to start your questioning. That's it. Make sure you read. You already know the activity, so make sure you answer your doctor's very well. Thank you.
Thank you.